Okay. Hello, everyone. Welcome to our third UBIC Chalk Talk of fall quarter. Today we have Dr. Graham Mcvicker presenting his talk, Discovering Novel Regulatory Sequences Through Analysis of Tiling CRISPR Screens. Dr. Mcvicker is currently an assistant adjunct professor of cellular and molecular medicine at UCSD. He received his PhD from the University of Washington, where he studied how mutation and selection shape human genetic variation. And he completed his postdoctoral research at the University of Chicago and Stanford about human genetic variation and chromatin state. Since 2016, he's been a part of the Laboratory of Genetics and Integrative Biology Lab at the Salk Institute. Dr. Mcvicker's research focuses on investigating how human genetic variation affects gene regulation through experimental and computational methods. He's interested in altering gene expression of immune and cancer cells to recognize regulatory variants. As part of this interest, his lab is developing new tools to discover regulatory mutations that contribute to cancer, including manipulating cell lines with CRISPR technologies. Dr. Mcvicker has also developed multiple tools and pipelines to analyze sequencing data, including WASP and BKGD. WASP is a software package for unbiased allele-specific read mapping and molecular QTL discovery, and BKGD is a program for calculating B-values across the genome. So please give a warm welcome to Dr. Mcvicker, and whenever you're ready, you can begin.
Okay, well, thanks very much for having me today. I'd like to keep the talk fairly informal, so I would encourage people, if they have questions throughout the talk, just to ask or raise your hand, and I'll try to get to you. And you know, I'm always happy to interact with the bioinformatics club. I know that UCSD bioinformatics undergrad program has a good reputation, and it's supposed to be quite a strong program. And it's been great working with undergrads in my own lab as well. So currently we have one computational undergraduate, Karthik. I don't know if he's a member of the bioinformatics club or not, but I think he's in data science and biology as a double major. But he's been really great in the lab, and in the past I've really enjoyed interacting with undergraduates in my lab. So if in the future you or somebody else is interested in coming by the lab to talk to me or other members of the lab, you would be welcome to just get in touch. Okay, so I will try to share my screen and get started with the talk. Okay, can everyone see it okay? Maybe somebody can just give me a thumbs up. Okay, great. Okay, so today I'm going to talk about work which we've been doing for several years since I started my laboratory, which is to use CRISPR screens to discover regulatory sequences in the human genome. My lab has actually what I would consider sort of 3 areas of focus. But it's all centered around trying to understand non-coding genetic variation in the human population and how it contributes to gene regulation. And we use different approaches to do this. So the approach that I'm mostly going to focus on today, let's see if I can do a laser here. Okay, is genome perturbation. So using CRISPR screens and various CRISPR technologies. But in the past, I've also done a lot of work. Hold on a second. I'm going to try to move the window. Okay, it's kind of getting in my way. Okay, but I've also used natural genetic variation a lot in the past. So in this type of approach, you actually sequence a number of human individuals, and you look at genetic differences between human individuals, and you can use these genetic differences to map what are called quantitative trait loci, which essentially are just genetic differences between people, which are associated with traits. And in my case, I was very interested in molecular traits, such as gene expression or chromatin. And there's still a part of my lab that is focused on this area, and we've been developing some methods for allele-specific expression analysis, and also using these to look for recurrent gene dysregulation in cancer genomes in recent work. Another area which we're more just starting to get into, but is to use machine learning techniques to actually predict variant function. So the idea is you can combine large amounts of data and large numbers of data sets, and without even doing experiments in the future, you should be able to predict what the function of a genetic variant is. at a molecular level. So that's something that we've become recently very interested in. But today I'm going to talk about mostly this genome perturbation area of what my lab works on. Okay, so I've divided today's talk into two parts. So in the first part, I'm going to talk about a method that we developed to analyze CRISPR screens. And in the second part, I'm going to talk about a CRISPR screen that we performed in my laboratory. So the idea of what we want to know in this part is we want to know if a region of the genome, which here I'm calling region X, has any kind of effect on the regulation of a gene, which might be located within, say, 100 bases or 1,000 bases or 100,000 bases or 1,000,000 bases of a gene that we're interested in. So how do we know if a region of the genome is important for the expression of a gene? And one way we can do this is with CRISPR technologies. So we can design a guide RNA that targets the Cas9 protein to this region. And then we can ask if, upon perturbing this region with CRISPR, does this have an effect on gene expression? And there's several different CRISPR technologies we can use to do this. So we can use standard Cas9, and what Cas9 does is it introduces a double-strand break. This typically gets repaired with non-homologous end joining. And this results in random mutations, 10 to 20 base pairs around the target. We can also use something called Crispr Interference, or CRISPR-I for short. This is similar, but instead of using an active Cas9, we use a deactivated Cas9. So this Cas9 doesn't have enzymatic activity. It doesn't cut the DNA itself, but it's fused to a repressive domain, such as CRAB. And what CRAB does is it results in heterochromatin formation in this part of the genome. So it silences this part of the genome. Kind of the reverse of CRISPR-I is CRISPR-A, or CRISPR activation. And in here, again, we have a catalytically deactivated Cas9. In this case, it's fused to an activation domain like BP64. And this results in histone acetyltransferases being recruited to this region of the genome and activation of what might be sort of a dormant enhancer sequence. And this can result in transcriptional activation. And another approach is that instead of using a single guide RNA, you can actually provide cells with pairs of guide RNA targeting nearby sequences. And when you do this, you frequently get deletions of the intervening sequence.
Similar to Perturb-seq?
Yes, so Perturb-seq is actually a flavor of these. The Perturb-seq is an experimental approach which could use any one of these CRISPR technologies. The difference with Perturb-seq, or what makes it a subset of these, is that it uses a single cell gene expression readout in the end, usually. And so the idea with Perturb-seq is that you can read out the effects of numerous genome perturbations on the expression of potentially thousands of different genes at once. But Perturb-seq is a flavor of this.
Okay, thank you. Does your lab use Perturb-seq at all, or?
We haven't performed what I would call a Perturb-seq experiment ourselves, but we are analyzing a Perturb-seq data set in my laboratory right now, a very large scale one that was performed in a different laboratory. So we are interested in Perturb-seq as a technique. I view it as sort of like a class of methods. It's essentially like a CRISPR perturbation coupled with a single cell readout is what I view as Perturb-seq as meaning.
Okay, thank you.
And what I'll talk about today, I'll talk about CRISPR perturbations with sort of what I would call bulk readouts, which means that you read out the effects with a selection or sequencing at the end on a population of cells, whereas with Perturb-seq, your goal is to read out the effects in single cells.
I see. Okay, thank you. That makes sense.
Okay, so I described how you can use this to look at just one sequence at a time, but ideally we'd like to look at thousands and thousands of sequences at a time. And so you can do this with CRISPR. So instead of using just a single guide, you can introduce thousands of guides into cells. And you can perform what I would call a CRISPR regulatory screen. So you design guides, for example, to tile across a region of interest. And then you can introduce those guides into cells. And each cell, you can design the system so each cell gets a small number of guides, perhaps 1 or 0 per cell. And then you can sort the cells which took up the guides into different pools. And you can do this either with a selection. For example, you could take only the cells that survive after some sort of harsh treatment, or you could look at proliferation. So you could see which cells expanded the fastest. Or you can sort them based on expression. So for example, you could sort cells based on the expression of a single gene into a pool with cells that have high expression of that gene and cells which have low expression of that gene. And once you've done this sorting or selection, you can actually reveal the guides which are present in these different pools by sequencing. So you can sequence the guides which got integrated into the genomes and just count how many times you see each guide in each pool. When you do this, you end up with a table like this, where for each guide, you have the counts for how many times you observed it in each pool. So in this example, we're counting how many times we observed this first guide in a low expression pool with 2 different replicates and in a high expression pool with 2 different replicates. And we look for significant differences in these counts to try to identify genome sequences, which were likely important for this trait that we're looking at. In this case, the trait that we're looking at is gene expression. Does that make sense to people so far?
Yeah, makes sense to me.
Okay, so our goal from analyzing these types of data are to identify regulatory elements. So sequences in the genome that are important for the expression of specific genes. And I'll start by giving an example from a published study that was published in 2017. And this is a CRISPR activation screen to try to find regulatory elements for this gene called CD69. And when they performed this screen in this paper, they densely tiled guides across this large region of the genome. And they did this experiment, which I described on the previous slide, where they sorted cells based on the expression of this gene, CD69. And when they analyzed the data from the screen, this is what it looked like. So they just computed a log-2-fold change of the counts from the high expression to the low expression pools for each guide. And this is what they use a sliding window to sort of smooth the results. And these are what the results look like. So you can see here I've highlighted in red the promoter for CD69. So we know this is a sequence which we know should control the expression of CD69. But beyond that, we're not sure what we should expect to see. And you can see that there's a number of regions which sort of stick out. But I would say that, like, just from looking at the data, which they provided in this paper, it's actually pretty hard to answer some basic questions about what's going on. So we'd like to know how many regulatory elements there are. And I think it's pretty hard to actually make a quantitative count just from looking at the results and where exactly they are. So you could make some educated guesses from this, but it's not really a sophisticated statistical method which they used in their paper to analyze this data. So we thought, there's a lot of room for improvement here, and we would develop a method to try to help more quantitatively answer these questions. When you have data like this, how many regulatory elements do we think there's statistical support for, and where exactly are they located? Are the basic questions that we want to ask. Okay, so here again, I'm showing the data from their paper. And I'm going to describe the algorithm which we developed to try to find regulatory sequences from data like this. So we start by segmenting the genome into small 100 base pair windows. And we label what we know are true functional sequences or regulatory sequences. So in this case, we're confident that the promoter of CD69 is a regulatory sequence for CD69. So we label it as such. So this is sort of our prior knowledge. And then once we label known functional sequences, we can estimate what we call sorting probability distributions, where we look at the guide frequencies in the different pools for the sequences which fall within this, for the guides which fall within this known functional sequence versus all the other guides, which we call the background. And we can estimate what the probability of your guide ending up in the low expression pool versus the high expression pool are from comparing these data. So here I've shown 3 different pools. So you can think of this as maybe low expression, medium expression, high expression. And you can see that the guides which overlap this functional sequence are much more likely to end up in the high expression pool than the guides from the rest of the sequence. So we can estimate these distributions from these data. And then once we've estimated these distributions, we can say, okay, now that we know what these sorting probability distributions look like for guides, can we identify another sequence which looks to be like a functional sequence, which sort of resembles this promoter sequence? And when we do this, we would predict this sequence here with some confidence looks like a functional sequence. And then we can re-estimate these sorting probability distributions with this new knowledge of this predicted functional sequence. And then we can identify another functional sequence. We can re-estimate these distributions again. And we can keep doing this iteratively until this process converges. Okay, so how do we know how well this method performs? It's actually really challenging. The reason it's challenging is because there's not really a gold standard out there of known regulatory sequences for really any gene in the genome. So for a lot of genes, people have known identities of a few regulatory sequences, but there's no sort of systematic resource for a gene like CD69 where somebody's systematically discovered every single regulatory sequence. In fact, that's exactly what we're trying to do using this type of technology. So how do we evaluate the performance? Well, one way to do it is we can conduct extensive simulations. So we can do a lot of simulations under a variety of different assumptions about how these screens perform and about the characteristics of regulatory sequences. And then we can see how well our method performs on various simulated data sets. So we did a huge number of different simulations with a number of different characteristics. And here I'm showing you a summary of the results. And I'm summarizing the results here in terms of a metric called average precision, which it's quite similar to computing the area under a precision recall curve, but it's a slightly better metric than that. And I'm comparing our method RELX to a competing method which was published while we were developing RELX, as well as a method called MAGIC, which it wasn't really developed for this specific type of CRISPR screen, but it was developed for CRISPR screens which targeted protein coding sequences in the genome. And here I'm showing you some different simulation parameters, such as how strong is the selection strength, and we think this is sort of a key parameter of these CRISPR screens, as well as how efficient are the guides at actually inducing these perturbations that we are interested in. And we can do a variety of different simulations with different parameters here. And I'm just summarizing a few of them. But you can see that in general, RELX performs much better than the other tools under a variety of different scenarios. So this is one way we can assess our performance. And based on a large number of simulations, we think our approach should work pretty well. We can also go back and reanalyze these published CRISPR screens. So remember I was talking about this study where they looked at CD69 as an example. And these are what the results from RELX actually look like running on CD69. So here we have predicted functional sequences here. And this shows what the real results look like from running on here. So these are the posterior probabilities that there's a functional sequence at each of these locations. And so we can also collapse these different tracks onto a single track where these are the predicted functional sequences of each type. And you can see, so we have some fairly nice clean predictions about where RELX thinks there are functional sequences for CD69. And here I'm going to compare these to these competing methods I mentioned, CRISPR, SERP, and MAGIC. And in my opinion, I think that our results look a lot more interpretable and cleaner than the results from the competing methods. And I can just to illustrate this a bit further, I'm going to zoom in on the small region surrounding CD69. So this is a zoom in showing a 20 kilobase region here. And we can see there are functional sequences that RELX predicts. So this is the training sequence that we gave it, that we call FS0. This is the CD69 promoter. And RELX predicts a number of additional sequences. So functional sequence 1, 2, 4, and 5 are all in the vicinity of CD69 in this region. And these are regions predicted by CRISPR SERP indicated with these yellow bars. And we can also do experiments to try to validate these predictions. And so this is just a negative control experiment where we're comparing what happens when we target, when we use a non-targeting guide. So it shouldn't be affecting the expression of CD69. And we can compare this result to when we target a positive control, so the promoter of CD69, and when we target the promoter of CD69 with CRISPR-A, as we expect, we see a big increase in expression of CD69. So these are all results from flow cytometry experiments. When we target functional sequence 1, we see again, this is a sequence predicted by relics, we see a big increase in expression. And when we target functional sequence 4, we again see a big increase in expression. So this is validating experimentally that these predictions made by relics appear to be true functional sequences. And when here's a prediction here, which was actually overlaps with the region predicted by CRISPR-Surf to be a functional sequence, but we do not predict it to be a functional sequence. And when we target this region, we see no difference from our negative controls. So this suggests to us that we think in this example, our experimental validations support that relics is doing a good job at predicting regulatory sequences from these data. Okay, since we published the first relics paper, we've actually been continuing to try to make improvements to the relics software. So we have a newer version, which we're calling relics plus. And it incorporates a number of additional features beyond what was in the first version. So it tries to better model guide efficiency. So it's known that different CRISPR guides have different efficiencies, meaning that some guides don't show efficient perturbations at all, whereas other guides do. And there's a number of different prediction tools that try to predict guide efficiency, and we try to incorporate their predictions into the relics software now. In addition, we have an improved what we call dispersion modeling. So it's known that count data in genomics is often what we call over-dispersed, meaning there's a lot more variation than you might predict given a standard statistical model for count sampling. But what's interesting about the data for these CRISPR screens is that we see that there's a relationship between the dispersion and the guide count, which is kind of nonlinear. And we try to model that now better than we did in the previous version. We also try to improve what we call the area of effect modeling. So it's known, for example, that when you have just standard Cas9, you tend to get indels or small insertions and deletions that occur mostly within 10 or 20 base pairs of your target site. Whereas with dCas9 CRAB and dCas9 BP64, so CRISPR-I and CRISPR-A, it's thought that you can exhibit perturbation effects up to 1 kilobase away. And so we try to model that directly now within relics. In addition, we've extended relics to be able to work with paired guide screens where we can have one guide targeting a sequence here and one guide targeting a sequence here. And some fraction of the time, they result in short indels around these target sites, and some fraction of the time they actually result in a deletion, for example, between the two target sites. And we model this with relics plus. And so these are the results from using relics plus on simulated data. And I want to emphasize that since we published relics, there's been an additional competing method published called MOD. And MOD actually works fairly well, but we think that relics plus works even better. And so this is results on some simulated data from relics plus. One of the things that we've used relics plus to analyze is a tiling deletion screen, which was performed for this gene OCT4. This was actually done in, this experiment was done in Bing Ren's lab at UCSD and published several years ago in 2017. And in their study, they validated a number of the sequences that they predicted. But here I'm showing you the analysis results from relics. So you can see these would be the predicted functional sequences from relics here versus analysis results from MAGIC or CRISPR-Surf, which really don't effectively identify any functional sequences from these data. And we wanted to look at whether the predicted functional sequences overlap with regulatory annotations. And so this includes sequence conservation, H3K27 acetylation, K4 monomethylation, a number of histone modification marks that are associated with active regions of the genome, as well as promoters. And what I'm showing you here are results from perturbing these sequence annotations. So this is sort of the null distribution. And what we actually observe by running relics plus in these software. So in all these cases, we see a substantial enrichment of these marks associated with the functional sequences that we identify around OCT4. We can quantify this level of enrichment, where this is the log2 fold enrichment, where 0 would be your expectation under the null. And if we can look at the features which were identified in the original paper using their analysis method based on magic. That's shown in this blue circle here. We can look at the ones which are identified by relics or the ones which are identified by both. And we've broken out these functional sequences by these different characteristics, and we show the relative enrichment of each of these subsets. And what you can see, I hope, is that the functional sequences that are predicted only by relics actually have some of the strongest enrichments for things like sequence conservation. So this suggests to us that we think that relics is actually doing a much better job than their previous analysis method that was used for this paper, because we see a much greater enrichment for the relic specific or the shared sequences than the ones which were only identified in the previous analysis. Okay, so that's the first part of my talk. In the second half, I'm going to describe actually a large experiment that we did to try to identify regulatory elements for GATA3. We analyzed this data using this relic software, which I described in the first part of the talk. Are there any questions in the meantime? I'd be happy to take a little break and answer any questions that people have on the first section before we go on.
Feel free to either unmute yourself or talk in the chat if you have a question.
And if you're shy, you can DM me the question. I'll ask it for you.
Okay. Well, I'm happy to move on if there are no questions so far, but hopefully people are following along. Okay, well, I'll keep going. Okay, so in this part of the talk, I'm going to describe a tiling CRISPR screen, which we did for this gene called GATA3. Now, GATA3 is known to be an important transcription factor. One of the things that's known to be important for is in the differentiation of T cells, which is one of the reasons that we're interested in it. But also just to motivate why you might be interested in GATA3 is if you look at this locus in the human genome, there's actually a large gene desert downstream of GATA3, where there are no other annotated genes. There's a number of annotated genes on this side of GATA3. But if you look at this locus, it's also has a huge number of risk variants associated with various human diseases. So in particular, there's a large number of genetic variants associated with different cancers. But there's also a large number of genetic variants associated with autoimmune and inflammatory diseases. And later on in the talk, I'm particularly going to highlight these associations with asthma and allergy. So it's an interesting gene for several reasons, but it's associated with, the region around it is associated with a large number of cancer and immune-related traits. And like I described, it's known to be very, very important and thought to be a master regulator of differentiation of T cells. And particularly, it's been studied in the context of differentiation of naive CD4 T cells into a highly specialized activated T cell subsets. So what I'm showing you here is just a cartoon of naive CD4 T cells. So these are helper T cells. And in their naive form, they're sort of just sentinels, but when they become activated, they can differentiate into these specialized subsets of T cells, which carry out various functions in the body. In particular, I've colored in Th2, the Th2 subset, darkest color, because it has by far the highest expression of GATA3. So here I'm showing you the expression of GATA3 in different immune cell subtypes, and with a particular focus on these subsets of T cells. So GATA3 is often thought of as a master regulator of differentiation of naive CD4 T cells into this Th2 helper T cell set. And Th2 cells are very important for defense against extracellular pathogens in your body, especially things like helminths, which are small worms. But it's also thought that Th2 cells are believed to be sort of overactivated in allergic diseases, such as allergy and asthma. Okay, so we wanted to better understand the regulation of GATA3. So we performed a tiling deletion screen around GATA3. So instead of using something like CRISPR-I or CRISPR-A, we used pairs of guides and used Cas9 to try to introduce small deletions into the genome. And we tile these deletions across the GATA3 region. And the idea behind tiling them is that we can have repeated deletions at each site. So this should increase our statistical power to detect effects. We can interrogate very large regions of the genome.
Oh shit.
What are you gonna do with them?
Probably eat them. I think, yeah, I'll probably eat them.
Okay. So to do this, we designed pairs of guides to tile these small deletions over a 2 megabase region centered on GATA3. And each of these pairs of guides was designed to delete about 800 or 1,000 base pairs of this sequence. So in total, we designed 15,000 guides to tile across this 2 megabase region of the genome. So in this experiment, we designed the paired guides to tile across the region. We had them synthesized into a pool of oligos containing these paired guides. We cloned them into a lenti-CRISPR V2 backbone. So this allows us to put these guides into a virus called lentivirus. And we can infect cells with the lentivirus carrying these guides. This is called transduction. So we transduce cells with the lentivirus carrying the guides. And then we do a selection so that we only retain the cells which actually took up the virus and integrated the guides into the genome. So after we have these cells which contain the guides, we can let the cells recover for a bit. Then we can sort the cells into different expression pools using an antibody targeting the GATA3 protein. We can then sequence the guide pairs which are present in each of these different expression pools and analyze the results with the RELIX software, which I already described. So overall, the workflow looks like this. We performed 4 different replicates of this study. And in the first 2 replicates, we just use a total of 4 pools. So this bulk or input pool, which is the unsorted cells, the high expression cells, the medium expression cells, and the low expression cells. In the 3rd and 4th replicates, we actually increased the number of pools. So we also had ones with very high, low, high, and so on. So to sort of fine-grained expression pools. And the first thing we did is we looked at whether we could see evidence that the screen was actually working. And so to do this, we estimated the sorting probabilities of guides into each pools. And we compared the sorting probabilities for guides which targeted GATA3 itself, so GATA3 exons versus everything else. And when we did this, we saw that guides which targeted the GATA3 exons were much more likely to end up in this low expression pool. And you see this sort of expected association where guides which target GATA3 itself are unlikely to end up in the high expression pool and likely to end up in the low expression pool. So this gives us some confidence that our screening procedure actually worked. Oh, okay, so there was a question by Alexandria, and she said, out of curiosity, how much did it cost to run this experiment? That's actually a good question. So, I think the synthesis of the guides itself, so that part of it where we designed the guides and synthesized them was not so expensive. So I would say we spent about $5,000 on that. But that said, the amount of work that's gone into this project is huge. And it's been years of work for a postdoc in my lab, as well as several other people who contributed to the project. So, and there's been numerous subsequent experiments for follow-up and so on. So I think when you just add up the reagents for the experimental cost, it's not so high, but the total amount of manpower invested into it is quite high. And actually, from doing a lot of science, that's where the dominant cost is, that you spend a lot more on people's salaries for performing the experiments and analyzing the data than you typically do on the experiments. So in total, I think like the reagents and sequencing costs and oligosynthesis costs for the experiment is not so high, maybe $20,000 or $30,000 in the end. But it's been years of work for people to work on the project. So in that sense, it's been, probably several $100,000 to do this whole study. Does that make sense? Okay, well, I'm going to continue on that. Okay, so these are what the results look like from actually analyzing the data with relics. So here I'm showing you the relic scores. And I'm showing you also some tracks of histone modifications, which are often used to identify putative regulatory sequences in the genome. I think something you'll immediately notice is that most of the functional sequences we identify, so here I'm showing you the relic scores, and then I'm showing you here the functional sequences with these dashed lines that are actually predicted by relics. The vast majority of the sequences predicted by relics are downstream of GATA3 in this gene desert region, with only 2 of them being upstream of GATA3. And I've highlighted 2 of them, functional sequence 17 and functional sequence 16, which I'm going to come back to in subsequent slides. Okay, and so the next thing we wanted to do is look at all these functional sequences which are downstream of GATA3, well, actually all of them total. So here, each column represents a different functional sequence predicted by relics. And then they're colored in by whether they overlap with or are near to these other sequence features. So here I've indicated ATAC-seq peaks. So these are open chromatin regions, as well as H3K27 acetylation peaks in GERCAT cells and H3K4 monomethylation peaks in GERCAT cells. So I'm indicating GERCAT cells because this is a cell line we perform the experiment in. And GERCATs are often used as a model cell line when studying T cells. I've also shown some data from naive primary CD4 T cells, which we obtained. And so you can see that there's a substantial amount of overlap with all of these sequence features. But we can actually quantify this level of overlap in terms of enrichment, again, using a permutation-based approach. And so here, this is the log2 observed over the expected, with sort of the null expectation being indicated by this line at 0. And you can see overall, we have a significant overlap with H3K27 acetylation and H3K4 monomethylation peaks. For direct overlaps, we actually did not see an overlap with open chromatin regions identified by ATAC-seq or DNA's hypersensitivity. However, we found that the functional sequences that we identify, while they don't directly overlap with these elements, they're often very nearby to them. So we see a substantial enrichment if we extend our functional sequences by a small amount. Oh, so there was a question saying that since we are working in bins of 100 base pairs, how or why are the functional sequences different sizes? That's a good question. So the relics algorithm actually allows several adjacent bins to be combined into a single functional sequence, if that makes sense. So you could have a functional sequence that was 500 or 600 base pairs long, because relics allows for the functional sequence to take up more than 100 base pair bin. So our interpretation of this result is that we actually think that relics may be a bit too aggressive with trying to pinpoint the exact location of the functional sequence. And due to the variability in the products that you get from CRISPR, sometimes you may get unintendedly longer deletions than you intend to. We think that sometimes relics predictions are actually a little bit off. So sometimes we think that they're probably not exactly overlapping the true functional sequence, but close to the functional sequence is our best interpretation of what these results mean. So we also looked at contact maps with H3K27 acetylation high chip data. So this is like a high-C experiment, which some of you may be familiar with, which tries to measure the contact frequency between things like promoters and other elements of the genome. And so what we see here is, here's the GATA3 promoter, which I'm indicating. We see that functional sequence 17, which I highlighted, definitely shows an increased contact frequency with that promoter. As does this functional sequence 16, which is a very distal functional sequence. And they also show an increased contact frequency with each other. So we wanted to sort of further quantify these contacts of all the functional sequences overall. And we computed interaction scores using this high chip data with the GATA3 promoter. And we compared non-functional sequences to functional sequences with these normalized interaction scores. And we saw that the functional sequences that we do predict do indeed show a substantially, on average, greater interaction with the promoter than the sequences we didn't predict. Okay, so the next thing we want to do is actually focus on several of these functional sequences. So we focused on functional sequence 17 and functional sequence 16, which showed evidence for contact with the GATA3 promoter. And we actually performed individual deletion experiments of these elements in GERCAT cells. And so this is a positive control here versus what we call a safe harbor sequence, which we don't think is functional. And we see a shift here towards lower expression when deleting the exon. We see a similar shift when deleting this functional sequence 17. And this sequence, I will say, looks kind of like a classic enhancer. So it shows a characteristic signature of what you expect an enhancer to look like, with a large level of H3K27 acetylation as well as H3K4 monomethylation. When we zoom in around the region containing FS16, we see that FS16 doesn't overlap any marks, but we do see that this element nearby overlaps K4 monomethylation. And like I mentioned on the previous slide, we think that RELX is actually sometimes occasionally missing the true functional sequence and actually is sort of predicting things that are nearby rather than directly overlapping. So we also tried deleting this, what we call primed enhancer 16 region here. And so I'm showing you the results from here. So in GERCAT cells, we actually don't see, when we delete this functional sequence 16, we don't see an effect as we would have hoped. And we see a very small, subtle effect when we delete this primed enhancer 16. So in this case, we think that we're probably mispredicting the true functional sequence with RELX. And in fact, the true functional sequence is probably this one here. Okay, so I want to revisit this idea of looking at trait-associated genetic variants, particularly around the GATA3 locus. So here I'm showing results from a genome-wide association study for allergic diseases. And when we analyze all of the genome-wide significant hits from this genome-wide association study, what comes up as the most enriched categories are all related to helper T cell differentiation. So we thought this was a good trait to look at because we're also interested in looking at T cell differentiation, and GATA3 is supposed to be a master regulator of this, and GATA3 is indeed, there's a GWAS hit near GATA3 in this study. If we zoom in on this hit that's near GATA3, we actually see there's two independent risk regions, which I'm going to label risk region 2 and risk region 1. So these are regions which, from genome-wide association studies of tens of thousands of people, are statistically associated with allergic diseases. And if we zoom in on this risk region 1, we can do what's called statistical fine mapping to try to predict statistically what are the most likely causal genetic variants. And it turns out there's 3 variants which are predicted to be each somewhat likely as being the true causal variant. And then when I zoom in on this region here, we see that 2 of them fall within this predicted regulatory element, FS16, and this other one falls within this nearby to it. So all 3 of them are close to this FS16 element that we identified with our screen. And I also want to emphasize that in naive CD4 T cells, this region here looks like a sort of primed enhancer, and that it has H3K4 monomethylation, but not H3K27 acetylation. But when we look at differentiated Th2 cells, this whole region looks like a very strong enhancer. Okay, so we actually performed deletion studies in primary Th2 cells. So we differentiated Th2 cells in the lab. And we performed these CRISPR deletion experiments here. And we can see that, yes, when we delete the exons of GATA3, as expected, we see a large reduction in GATA3 expression, as you would hope. You see a similar reduction when you delete the FS17 element. And when you delete FS16, the lead SNP and this part of this paired enhancer or primed enhancer 16 element, these all result in decreases in GATA3 expression. So to us, this indicates that, yes, this region is very important for driving GATA3 expression in differentiated Th2 cells. So that's all the data that we have so far. So I'll just conclude the talk, and I'm happy to have a few minutes of discussion or questions at the end. So today I told you about the RELX software, which is powerful for several reasons. It can jointly analyze multiple pools all at once. It accounts for things like the area of effect, dispersion, and differences in guide efficiency, the new version of RELX. It outperforms all the other methods which have been published. And we think we can use it to identify novel regulatory elements. We also talked about a tiling deletion screen that we performed to try to systematically identify sequences that affect GATA3 expression. And something that's interesting that I didn't really focus on that much in today's talk was that we see that many of the sequences overlap this H3K4 monomethylation mark, but not the active enhancer H3K27 acetylation mark. And what we think this means is that these are classically associated with what I would call primed enhancers, which are sort of ready to be activated, but not yet activated in that cell type. But we think that deletion of these primed enhancers actually does result in subtle changes in expression, which are detectable with our screen. And many of these so-called primed enhancers do become activated in the activated helper T cell subsets, like the Th2 subset that I described. And I just want to thank the members of my lab. So many of them contributed to this project. The GATA3 screen in particular was led by Shoei Chen, who's a postdoctoral scholar in my lab. And the Relic software was developed by Patrick Fiao, who was a BISB PhD student who defended in the winter. He defended in February. But many other people also contributed in different ways to this project. I'd like to also point out Karthik, who's an undergraduate at UCSD, who contributed a lot to the development of the Relics Plus and the results that I showed today. Our collaborators in Bing Ren's lab also assisted a lot with setting up our GATA3 screen. And these are just a couple of photos, recent photos in the past year of our lab. So like most people, we did a lot of Zoom in the past year. But when the case counts were lowest, we actually had a couple in-person events as well. Okay, so with that, I'm happy to stop, and I have about 10 minutes where I could take some questions if people are interested. And I'm also happy to answer, any kind of questions you may have. They don't necessarily have to be related to the talk that I gave today.
Again, if you have a question, just feel free to either unmute or you can send it in the chat or privately message either me or Anjali, and we can read it out loud.
Well, maybe I'll ask you a couple questions then. So how many students are members of the UBIC club?
Actually,
Any actual membership? Oh yeah, that's we do.
So we have one question really quick from Ishan. He asked what kind of model is Relics based on?
So I would call it a Bayesian hierarchical model. At its core, the distribution that it uses to model the sorting probabilities of the guide counts into different pools is what's called a Dirichlet multinomial distribution. So it actually models the proportion of guide counts which end up in each pool for guides which are functional or target a functional sequence versus those that do not target a functional sequence. Does that make sense?
Yeah, that does make sense. So maybe I'm just lacking a bit of background on the Bayesian hierarchical model, but why exactly made you choose that kind of model? Is it to do with some theoretical nature of the guide RNA or some previous literature?
Yeah, that's a good question. So, there's a number of analysis choices you could make for how to analyze the data. For me, I like to think about modeling usually in a statistical way. So we tried to motivate what we did statistically. And from prior experience working with count data, I knew that some sort of count-based distribution would likely be best. And so we actually tried, several different approaches. So one approach might be to think of it as, you could think of it as just Poisson-based sampling for counts. But in practice, when you analyze genomics data, the Poisson distribution almost never accurately describes count data. And instead, people frequently go to what's called a negative binomial, which is, you can think of as an over-dispersed Poisson distribution. So that was one choice we could have made, but we found it a little bit, I guess, easier to model or think about in terms of thinking about proportions of a guide that ended up in each pool, in which case that would be better modeled with a multinomial distribution or a binomial distribution if you had two pools. But we again found the data to be quite over-dispersed, which meant we went from basically a multinomial to what we call a Dirichlet multinomial distribution. And the Dirichlet multinomial distribution, again, allows for over-dispersion. And then we had to make it a little bit more complicated because rather than just having a single dispersion parameter, which we had in our original version of Readouts, we found it actually better to allow the dispersion level to vary with the total guide count for the guide that we were looking at. And so we allow that now. So we have a function which describes the relationship between dispersion and guide count. And some of the other features are sort of more based on intuition behind sort of how the technology works, like the area of effect modeling, for example, where, and the fact that we want to allow for multiple guides to potentially affect a single functional sequence or sequence in the genome. So that's all sort of motivated behind how the thing works. Does that make sense?
Yeah, that does. Yeah.
So it came, there's like a statistical motivation behind like the distributions that you choose to use. But then there's like, how you put it all together also has to make sense in terms of like the spatial organization of where the guides are targeting. And then, we tried a number of different things and then chose one that seemed to work, but they all started with some sort of intuition about why it should work.
You said the model does give intuition on why that works, or?
Well, I mean, we have some intuition based on like the structure of the data about what model what kind of models we think should work.
Right, because you can look at the actual values and see kind of which distribution would.
Yeah, I mean, at the most basic level, what you have are counts for each guide in a big table, right? And when I see count data, I immediately think, well, we should use some sort of count-based statistical distribution. And so we can start there, but then add complexity as needed.
Right. Thanks. Have you looked into like using some other kind of models though, or like do you have any future insights or future directions on the modeling side of things, like modeling maybe CRISPR efficiency or other gene editing ways for like talons and ZFNs and other stuff?
Yeah, I mean, I do.
Or is your focus more on the gene analysis part now?
I mean, I always am aware of there being like the possibility for improving relics more. I think there's a trade-off though, because it's sort of like, well, do we invest effort into that versus, trying to advance some other area of what we're working on. So there is kind of a trade-off of like sort of diminishing returns of continuing to develop the same model versus, trying to do more or different things or extend it in different ways. I think Aditya mentioned at the beginning Perturb-seq, which is more like a single cell based approach. And so one of the things we are actively doing is use a model which has some similarities to relics, but apply it to one of these single cell data sets. So we're taking some of the lessons we learned doing relics and trying to apply it to a very large single cell data set.
Right.
And that's something actually that Karthik has been working on in the lab, and he's an undergraduate actually.
Yeah, I actually happen to know Karthik. Yeah. So what exactly is the difference between bulk RNA-seq and single cell RNA-seq? I mean, I understand single RNA-seq. Is bulk RNA-seq just the general RNA-seq workflow? Is that
Yeah, bulk RNA-seq is just the original RNA-seq.
okay, cool.
And
Yeah.
so in that case, you might have 100,000 or 1,000,000 cells in a mixture. And you can sort of think of the sort of classic way people describe it now, I think, is like as a smoothie. It's like you throw everything in a blender and then you get the results. Whereas with the single cell approach, you try to get the individual component results, right, of the things that would go into the smoothie. So you try to get measurements per cell. And so you might have an experiment where you have 20,000 single cells and you have measurements for every gene of the expression level in those single cells. There's a trade-off though, because with the single cell data, it actually tends to be quite sparse and noisy. So you have a sort of limited number of counts that you get per cell. And as a result, your measurements in a given cell are very, very noisy. And so there's been a huge array of different methods people have tried to develop to better analyze single cell RNA-seq data.
Right.
So it's cool because you do get measurements per cell, but it's challenging because your measurements per cell are very noisy. And so in the end, people often end up doing some sort of clustering or aggregation of the data.
Right.
Okay, well, thank you everyone for the opportunity to come talk to you today. I'm going to have to go because I have another meeting right after this that I have to attend. But if any of you have further questions, you're welcome to get in touch with me. I would recommend using my Salk email address, gmcvicker@salk, rather than my UCSD one, because I check that one a lot more frequently. And then I'm always happy to chat about science or working at a Salk and so on.
Thank you very much for coming and speaking.
Yeah, thank you very much.
Send out your email to everyone on the Discord so that they have your contact.
Okay, yeah,